our recent study that was, to be presented at the Alzheimer's Research UK conference was focused on aspects of the local expression of brainwaves in sleep and their relationship to pathological markers in older adults. Now, we wanted to look in older adults that did not have mild cognitive impairment and did not have Alzheimer's disease, but instead had a family history of AD, because we wanted to look early in the process, because we wanted to understand how early can you see sleep disturbances associated with Alzheimer's pathologies. And we did this because we wanted to see and answer the question, can sleep disturbance be present even in older adults that are at risk for Alzheimer's disease, but don't even have amyloid plaques yet, right? And so we looked at different brainwave features that have been shown in other studies later on in the process to be disrupted by Alzheimer's pathology. And we looked specifically at brainwaves that are shown to actively promote memory retention over long term. So these are brainwaves that are known to be critical for memory. And these two brainwaves are called slow waves and sleep spindles. They're defining oscillations of sleep in non-REM sleep in particular, non-rapid eye movement sleep in particular. And we know that they're disrupted in age. We know that they're disrupted in different ways in Alzheimer's disease. So we brought, this is a collaborative effort across several groups, our group, a group in Wisconsin, and a group in Sweden, and a bunch of people who were part of the Wisconsin-Madison's ADRC, Alzheimer's Disease Research Center, came in, and they had kind of serial regular longitudinal spinal taps where their cerebrospinal fluid was assessed. In addition, they came in for a polysomnography, which is the classic gold standard way of measuring sleep. But in addition to looking at the basic measures of polysomnography, which includes EEG electrodes, EMG, and EOG to look at eye movements, muscle movements, brainwave activity, clinical signals of respiratory breathing to look for sleep apnea, for example. In addition to that, our EEG was actually a high-density EEG net that had 256 electrodes. So we had a very good resolution to look at the local expression of these brainwaves that are memory relevant. And so what we decided to do is look at them and relate them to a series of Alzheimer's disease fluid biomarkers that you can see in the CSF. Are they related in this very early cohort? And a lot of which may never get Alzheimer's disease, but some of which definitely will. So we looked at that, and we examined how the expression of these slow waves and spindles are related to CSF measures of amyloid, CSF measures of tau pathology, as well as fluid biomarkers that measure other things that are important, that are thought to be important for the Alzheimer's disease process, including inflammatory markers, such as the marker YKL-40, which is a marker of glial activation in the brain, which is critical in the process, and it gets overactivated in Alzheimer's disease. We also looked at markers of neuronal integrity, including neurofilament light protein, which is associated with axonal degeneration, so degeneration of the part of the neuron that connects and communicates with other neurons and sends signals to other neurons. And we also measured marker neurogranin, which is associated with synaptic density. Or synaptic degeneration. This is a marker that is associated with degeneration of the connections between different neurons. So we can look at all of these markers and we can say, are they related to expression of sleep in any way? Are they related to each other? And one of the central questions we wanted to know is something that hadn't really been explored that much, which is age, the effects of age on inflammation, triggering a process of pathology and triggering a process of degeneration, which ultimately impacts sleep expression even prior to amyloid plaque deposition. And so we found, in fact, that was the case. We found that age was predicting both inflammation and also axonal degeneration or NFL buildup, and that both of these markers were predicting the expression of amyloid or affecting the expression particularly of tau, actually, in phosphorylated tau, total tau and phosphorylated tau. And both of those were predicting disruption and deficits in the expression of sleep spindles, which are an important oscillation, as we told you, for neuroplasticity and for memory functions. In addition, some of the ability of these tau pathologies to affect spindles depended on their effect on synaptic integrity. So in other words, we were able to build a tree of evidence suggesting that age impacts inflammation in the brain, inflammation impacts the buildup of tau proteins, and those impact the neuronal integrity, connecting different neurons, and ultimately disrupting the ability of those neurons to express functionally sleep oscillations that trigger neuroplastic events in the brain and foster sleep-dependent memory. So this is the overarching finding, and we think this is important because it builds on prior work and shows that, in fact, there are different mechanistic links, in pieces between the Alzheimer's pathologies and the sleep disruptions, and that it is actually related to inflammatory mechanisms in the brain and related to the degeneration of neuronal integrity itself.